Dear students, let us continue our study in this paper entitled Drugs of Abuse. In the previous module, we have learned how the analysis of acidic drugs is done, and you became familiar that what are acidic drugs and how their analysis is done. In this very particular module, we shall focus our study on the basic drugs. And if we try to look at the learning outcomes of this module, after studying this module, you shall be able to know what are basic drugs. You shall be able to learn how the preliminary examination of these basic drugs is done, and you shall be able to identify the alkaloids and active constituents of these basic drugs. Now let us begin with knowing what are basic drugs. Just very analogous to acidic drug, here again the name is very clear that these are the drugs which contain basic groups in them, and obviously they are alkaline in nature, and when they react with acids they will be forming salts. So with this background, let us go further. So we can define drugs which are alkaline in nature as the basic drugs. And if we talk about their structure, they contain different moieties having ring structure, for example pyridine, pyrrole, isoquinoline, quinoline, which contain heteroatoms like nitrogen, sulfur, oxygen in these rings. And these compounds, they react readily with acids forming salts. If we talk about their classification, dear students, their classification is based on their physiological action they produced, and if we talk about these physiological action, they include their narcotics effect, the deliriant, they are acting as spinal drugs or used as the cardiac drugs, and so on. So let us talk about all these in more detail. Talking about narcotics, these are the drugs which interact with those receptors in the brain which are responsible for the transmission and response to pain, and they are also called as narcotic analgesics or simply narcotics. For example, if we talk about the opium alkaloid, both natural as well as synthetic, morphine, codeine, monoacetyl and diacetyl morphine. So these are all examples of these drugs. On the contrary, the peripheral analgesics, for example aspirin, they have no abuse potential. Talking about deliriants, these drugs, they affect the vital part of the brain, and under this category come the atropine, hyoscyamine, cocaine, muscarine, which are obtained from fungi, mushrooms, and so on. Next are the spinal drugs, and as the name suggests, these drugs affect the spinal tract. For example, strychnine, brucine, and so on. Coming to cardiac drugs, these are the drugs which affect the functioning of heart, and under this we have quinine, aconite, nicotine, digitalis glycosides, and so on. Now talking about their identification. The identification of these drugs is of prime importance in clinical toxicology. When these drugs are consumed by the user in more therapeutic dose, the outbreak of signs and symptoms occur, and the drugs are likely to be distributed in blood as well as urine. Thus, in clinical toxicological cases, blood and urine are the main crime exhibit, and in acute cases resulting death, the stomach contents and the viscera which are to be taken into consideration for extraction of drugs along with blood or urine to establish and provide evidence that yes, this is the case which contained this particular drug. and because of this the poisoning or the death which has happened. So, let us try to understand how the extraction of basic drugs from biological matrices is done. And here again, just like the treatment we had done for acidic drugs, here again the drug has to be extracted from these biological samples with the help of solvent extraction, that is the solvent extraction method. And if you recall, a solvent extraction method consists of a system of two immiscible liquids which is required for the separation of material, and that is done by solvent extraction. The active constituent should be unevenly soluble in the system, thereby facilitating the extraction of the constituent from one phase to the other. In very simple terms, you can say that if it is soluble in one particular solvent, the other we are adding, that should be able to extract that very particular component in itself, leaving the other components, and then that particular solvent can be removed and concentration can be done. So, that is what is done in solvent extraction. And if we talk about the efficiency of the extraction, it is determined by the distribution coefficient, and in this case, like the earlier also, the distribution coefficient D is defined as total weight in grams of the solute in the organic phase divided by the total weight in grams of solute in the aqueous phase, or in general you can say in phase 1 divided by phase 2. If one of the two liquids contain a solute, this method is found more suitable. The system in this case is first shaken and then it is allowed to settle, and some of the solute is then transferred to the other liquid. Each of the liquid in a mixture of two immiscible liquids of this kind is referred to as a phase. Thus, some of the solutes are transferred from one phase to other phase, and that is what will be taking that very particular drug into the other phase, which can be then taken out. The amount transferred depends upon the relative affinity of the solute for each of the two solvents, which is referred to as the relative solubility, and this is determined by D. Greater is the value of D, greater is the efficiency of the extraction. The immiscible system may involve two organic solvents. The extraction for this system may be impaired due to the formation of emulsion. So, this you have to keep in mind, and so solvent extraction method has now been upgraded and made automatic with the use of accelerated solvent extraction technique. So, let us now study the Stas Auto method, and this is very similar to what we have studied in case of extracting the acidic drugs in the previous module, but there is a difference because there it was the acidic drug. So, we use the bases to extract, and here because it is basic drug we will use the acid. So, let us see the procedure here. 50 grams of biological material is minced and mixed with plenty of rectified spirit in a flask and then acidified with tartaric acid. After this, the mixture is heated on the steam bath for 1 to 2 hours with thorough shaking at frequent intervals. The extraction is then allowed to proceed for about 24 hours with steam off. The mixture is then filtered by using filter paper, and the filtrate is evaporated and the residue is again extracted with alcohol. All the filtrates are then combined in one porcelain basin. To the final filtrate, you have to add 2 ml of ammonia and then transfer to a separating funnel and extract with a suitable solvent like ether, hexane in portion of about 25 ml of each lot. This step of extraction is repeated 2 to 3 times, and the solvent would take up the basic drug and its active constituent. The basic aqueous solution is then used. If morphine is suspected, it may be extracted at this stage by amyl alcohol or chloroform to ether in 3:1 ratio mixture, or chloroform is to alcohol in 9:1 ratio mixture. Of these, amyl alcohol is the best, but as it is prone to form annoying emulsions, the chloroform to ether mixture is used as a preferred solvent system. The combined chloroform or amyl alcohol extract, depending upon whatever you have used, they are then further evaporated to dryness, and the residue is now ready for further purification and analysis. The evaporated chloroform extract is purified by dissolving it in about 20 ml of water, acidulated with sulphuric acid, and filtering through a small filter. The filter is extracted with chloroform first in acid and then in alkaline medium as the initial stages of extraction. These extracts are then further evaporated to dryness for analysis. Let us now understand the ammonium sulphate method, and this method is most useful for preliminary analysis or screening of the basic drugs like alkaloids and tranquilizing drugs, and also identification and semi-quantification. The visceral material, about 100 grams, they are cut into pieces, macerated and mixed with 100 ml of 5% acetic acid and then taken into 600 ml beaker. Solid ammonium sulphate is then added to it by frequent shaking to make a saturated solution. About 20 grams of solid ammonium chloride is then added in excess. The mixture is then heated in a boiling water bath for around 3 hours, and for suspected poisoning by aconite, the temperature you have to take care should not exceed 60 degrees. The mixture is then cooled slightly and filtered through the filter paper. The residue on the funnel is again extracted with 2 portions of 100 ml of 5% acetic acid and filtered as earlier. The filtrates are then combined and taken into a 500 ml separating funnel. The ether fraction is added to the aqueous acidic extract in the separating funnel and shaken for 5 minutes and separated. 100 ml of ether is again added to the acidic layer, shaken for 5 minutes and separated, and all these ether layers are then combined. The ether layers, which is the acid ether layer, and notice that this acid ether layer will be taking the basic compounds in them. After washing, the sodium carbonate is then extracted twice with 25 ml portion of 1 normal sodium hydroxide solution. The aqueous layer are separated from ether layer, combined and taken into another separating funnel. The aqueous solution remaining in the separating funnel after separation of acidic drugs is then made alkaline by addition of ammonium hydroxide and extracted 3 times with 100 ml portions of chloroform ether mixture, that is 1:3 ratio, and the aqueous layer is retained for extraction of opium alkaloids. So, this is how this extraction is done, and further tests can be done on these. So, let us now understand how the analysis of basic drugs is done. And first of all, let us understand the screening test, and how this is done is, to the residue of the extract that you have obtained after the solvent extraction, to this residue a few drop of Mayer's reagent is added. And if we talk about how the Mayer's reagent is prepared, it is prepared by dissolving 1.357 grams of mercury chloride and 5 gram of potassium iodide in 100 ml of water. And when you have added this few drops of Mayer reagent to the residue of the extract, and if you find that there is a white or gelatinous white precipitate, then it will infer or indicate the presence of basic drugs. To another portion of the residue that you have obtained after solvent extraction, add a few drops of phosphor tungstic acid. If there is a formation of red precipitate, it will give you indication that that extract is containing the basic drugs. Now, you can also add some other reagents, that is, and specifically as you can see in this table, the alkaloid, if it is morphine, the color which is developed with the reagent which is Marquis reagent will be violet. If you add conch nitric acid, there will be bright orange yellow color, and if you add the Mendelian reagent, there will be dark reddish brown color. On the other hand, if the biological sample is containing heroin as the alkaloid, then Marquis reagent will give you reddish purple color, conch nitric acid will give you pale yellow color, and Mendelian reagent will give you reddish brown color. If the alkaloid is codeine, Marquis reagent will give you dark violet color, conch nitric acid will give you greenish yellow color, and Mendelian reagent will give you olive green color. So, that is how you will be able to get inference that whether the substance which is present as the basic drug in the biological sample is morphine, heroin or codeine. Let us try to know some more color tests, which we can call them as special color tests, which are performed on the extract for detecting these basic drugs. If the basic drug being suspected is strychnine, what reagent you have to use? You have to take potassium dichromate and add conch sulfuric acid. So, that is what is the reagent. And how to prepare this? A crystal of potassium dichromate is then drawn by means of a glass rod through the sulfuric acid. So, this resulting is then added to the extract. The color observed will be, it will be a play of color which will be observed. There is a momentary blue which will be changing to a beautiful violet color, which then gradually changes to reddish purple, red or orange, and finally to yellow. And the color is discharged by adding stannous chloride, but it reappears on adding conch nitric acid. So, if you perform this test, just be very vigilant that what are the color changes taking place, and if they happen to be in this sequence, then it will indicate that it is the strychnine which is present as the basic drug. Coming to brucine, the reagent used here is conch nitric acid. So, if you add conch nitric acid to this extract and if you get red color, it will indicate that brucine is there as the basic drug in that particular sample. Let us continue with more color test by using some more different reagents. Various basic drugs, they respond to different reagents to produce a variety of colors, and this can be useful as a screening test. And you can see here in this table that how different reagents, when they are added, there will be formation of different colors, and based on that color you can have some inference about the class of compound which may be present. For example, if we talk about the reagent which is formaldehyde sulfuric acid reagent and how it is prepared, you have to take 4 parts of sulfuric acid and add 6 parts of formalin. The color observed, if it is red, pink, blue, violet, red violet or blue violet, that is ranging from red to blue violet, then the compounds which may be present as the basic drug in the sample will be benzodiazepines or phenothiazine. Coming to another reagent, it is the FPN reagent. And how the FPN reagent is prepared? 5 ml of ferric chloride solution is taken. To this 45 ml of 20% weight by volume solution of perchloric acid is added, and 50 ml of 50% volume by volume solution of nitric acid is added. So, these 3 are mixed, you will be getting the FPN reagent. And when you add FPN reagent to the extract, and if there is production of orange red, violet red, brown red, orange, red orange, pink orange, blue violet or red brown, then this will be indicating the presence of phenothiazine. Coming to another reagent, which is iodoplatinate solution. How the iodoplatinate solution is prepared? You have to take 2 ml of 5% solution of platinic chloride in 2 normal HCl and mix it with 5 grams of potassium iodide and then add 98 ml water with stirring. The resulting is the iodoplatinate solution. And when you add this to the extract, and if you observe violet, blue violet, brown violet, grey or violet color, it will be indicating that the alkaloid is present. Coming to Kopany-Zwicker test and the reagent, how it is prepared. 1% solution of cobalt nitrate in ethanol is prepared, and after adding above reagent, 0.1 ml of pyrrolidine is added. The color that you observe, if it is violet, it will indicate the presence of sulfonamides as the basic substances as drugs. Coming to yet another reagent is the forest reagent. Mixing together equal volumes of a 0.2% solution of potassium dichromate, a 30% volume by volume solution of sulfuric acid and a 20% solution of perchloric acid and 50% solution of nitric acid. Mixing all these, you get this reagent, which is forest reagent. And when you add this to the extract, then if you observe red, violet red, brown red, orange, pink orange, red orange, brown or blue color, then what it will indicate is the phenothiazine or imipramine and related compounds. Coming to Marquis reagent. How the Marquis reagent is prepared? It is prepared by mixing 1 volume of formalin and 9 volume of conch sulfuric acid. If there is formation of yellow orange color, it will indicate benzodiazepines. If there is formation of violet color, it will indicate morphine. If there is formation of reddish purple or dark violet color, it will indicate the presence of heroin or codeine. Coming to conch nitric acid. Conch nitric acid will contain 69 to 70% weight by weight of HNO3. Usually it is already ready made prepared in bottles labeled as conch HNO3. So if you take that and add it to the extract, and if there is formation of bright orange to yellow color, it will indicate morphine. Pale yellow color will indicate heroin, greenish yellow color will indicate codeine and red color will indicate brucine. Yet another important reagent is the Mendelin's reagent. And how the Mendelin's reagent is prepared? Dissolve 0.5 grams of ammonium vanadate in 1.5 ml of water and dilute it to 100 ml with sulfuric acid, and then filter the solution through glass wool. Resulting will be the Mendelin's reagent. And when Mendelin reagent is added to these extracts, if there is formation of dark reddish brown color, it will indicate morphine. Reddish brown color will indicate heroin. Olive green color will indicate the presence of codeine. Coming to cobalt thiocyanate reagent. In order to prepare this cobalt thiocyanate, you have to take 2.5 grams of cobalt thiocyanate and mix it with 16% HCl. When you are adding 1 drop each of these two, that is the cobalt thiocyanate and HCl, if there is formation of blue color on adding 1 drop of each of these reagents, then it will be indicating the presence of methaqualone, PCP, cocaine and heroin. Coming to the antimony pentachloride reagent. How that is prepared? It is prepared by passing chlorine gas through dried or anhydrous, which is made anhydrous by P2O5, and molten antimony trichloride and dissolving it in chloroform. If you get green brown or brown turning to black violet, then it will indicate the presence of beta-methasone, dexamethasone, digitoxygenin, desoxygenin, digitoxin, disoxin, and lenotoside C as the basic drugs to be present. Coming to the copper sulphate solution or the reagent, it is prepared, it is the 1% solution of copper sulphate in water. And if it is added and you have the observation of orange brown to green color or brown violet brown color, then it will be indicating the presence of sulfonamides. Coming to the mercurous nitrate reagent, how the mercurous nitrate reagent is prepared? It is prepared by adding solid sodium bicarbonate to a saturated solution of mercurous nitrate. A precipitate will form here which changes to a biscuit brownish color, and this should be used always freshly prepared. And if there is formation of a dark grey or black color, then it will indicate that the sulfonamides with having additional rings in them are present as the basic drug. Coming to yet another reagent, which is the Dresendorf's reagent. Here, in order to prepare this reagent, 1 gram of bismuth subnitrate is dissolved in 3 ml of 10 molar hydrochloric acid, and if necessary some gentle heating has to be done. It is then further diluted to 20 ml and it is dissolved in 1 gram of potassium iodide. If there is black precipitate of bismuth triiodide which separate, it has to be further dissolved in 2 molar hydrochloric acid. And taking this reagent and adding to the biological extract, if you obtained orange, red orange, brown orange precipitate, then it will be indicating the presence of alkaloid which may be primary or secondary or tertiary amines in their constitution, and they will be present as the basic drugs. After these tests, let us also try to know the special color tests which are used for different drugs of abuse. These are cobalt thiocyanate test, where the reagent is made by mixing these two, that is 16% hydrochloric acid with 2.5 grams of cobalt thiocyanate in 100 ml of water, and 1 drop of each of these solutions will be added, and you have to notice the corresponding test. Another reagent that you should prepare, and then I will show you the color chart that you can use to identify which compound it will be indicating. That other reagent is Fisher-Morris reagent, or Fisher-Morris test is the name given to this test. Here again we take two reagents, one which is conch formic acid, the other is 5% aqueous sodium nitrite solution. And how this is performed? You take 5 drops of the concentrated formic acid solution and 3 drops of the 5% aqueous sodium nitrite solution, they are added to the sample one by one. And after 2 minutes, 15 drops of chloroform is added, and color which is obtained in the layer of chloroform is then observed. So, this table, as you can see here, is giving you now idea that if you have performed the cobalt thiocyanate test based on the color observed, what compound it will indicate to be present, or the color observed in Fisher-Morris test in the aqueous layer and chloroform layer. As you can see here, if the compound is methaqualone, the cobalt thiocyanate test will be giving you blue color. In aqueous layer of Fisher-Morris test you will get no color, but in the chloroform layer you get the yellow color. Similarly, if the compound is cocaine, the cobalt thiocyanate test is giving you blue, the Fisher-Morris test, the aqueous color is giving you faint green and chloroform layer is also giving you faint green. Similarly, you can notice the different colors for phencyclidine, which will be blue, tinged respectively in this sequence. Caffeine, it will be pink, no color in the same sequence of these tests respectively. If it is heroin, it will be blue in thiocyanate test, but it will be faint blue in aqueous layer of Fisher-Morris test, and it will be yellow in the chloroform layer of Fisher-Morris test. If it is diphenhydramine, then blue color for thiocyanate test, yellow in aqueous layer and no color in chloroform layer for the Fisher-Morris test. If it is diazepam, then it is blur green in thiocyanate test, it is faint yellow in aqueous color in aqueous layer and faint yellow in chloroform layer of the Fisher-Morris test. So, this is how you will be able to differentiate by performing these tests. And again here, if you ever have some doubt, let me tell you here, you can always take a known sample as reference sample and perform the same test. It is always advisable to do that, because clearly you have the results in front of your eyes and you can simply compare that. If it is matching each and every test, then and only then these two compounds are same. And the next procedure or the next technique you can use for further identification and confirmation is the use of TLC, that is thin layer chromatography. Let us now understand how the identification of basic drugs is done by TLC. TLC conditions, if we try to see for the opium alkaloids, the TLC plate used is silica gel G with 0.2 mm thickness. Development is done by ascending technique, and the solvent system we have methanol is to ammonia in 100 is to 1.5 volume by volume. If we look for the spray reagents and the corresponding color of spots, if we take the iodoplatinate solution, which we have studied how we have to prepare this, the same spraying reagent can be used, and the colors that you have to look for are violet, blue violet, brown violet for alkaloids, and it can be even grey violet spots. You can also use the Dragon-Doff's reagent that we have learned how to prepare, and if you use that, then there will be orange spots for alkaloids and benzodiazepines. If you use the FPN reagent as the spraying reagent, then the colors you have to look for are orange red, violet red, brown red for the phenothiazines. Similarly, you can have orange red orange or pink orange, blue violet, all these will be indicating that the alkaloid is present. And yes, do not forget the extract of sample along with the control drugs and the known alkaloids you have to place, and if everything is matching, then it will be indicating that it is the same drug, whether it is the RF value or it is the colors of the spot which will be coming by using different spraying reagents. Let us look at the TLC conditions for some amphetamines, other stimulants and anorectics which have to be used. The plate here is almost the same, that is the silica gel G with 0.2 mm thickness. Development here also is by ascending technique, but when we look for solvent systems, they can be 3 solvent systems. So, the first solvent system is methanol is to ammonia in 85 is to 15 ratio. The second solvent system you can use cyclohexane to toluene to diethylamine in 75 is to 15 is to 10 ratio, and the third solvent system you can employ is the chloroform is to methanol in 90 is to 10 ratio. The spray reagents which are used are Dragon-Doff spray and acidified iodoplatinate spray. Using the Dragon-Doff spray, we have seen how this reagent is prepared. So, that can be used as a spraying reagent, and you have to observe for yellow, orange, red orange, brown orange spot, which will be indicating the presence of these substances. If you use the acidified iodoplatonates as the spraying reagent, then look for violet, blue violet, grey violet or brown violet spots, and calculate their RF values and always compare with the literature value which is available, and also always the standard reference of the known compound will be always a handy help to you. If everything is matching, that is the RF in that particular solvent system along with the color in different spraying reagents, then and only then the two substances are same. So, TLC that way is an excellent technique which can help identify it, and that too in very minute quantities, because when you are getting the biological samples, the quantities of the drugs will be in very small amount. So, all these tests and the TLC will be helping you identify which particular basic drug was consumed and is present in that biological specimen, and it will be helpful in solving that particular case. So, dear students, let us now summarize what we have learned in this module. We have studied about the basic or the alkaline drugs. Basic drugs, as the name suggests, these are basic or alkaline in nature, and when they react with acids, they will be giving you salt formation. And if you look at the various types of basic drugs, we have studied they are narcotics, stimulants, cardiac drugs, deliriants and spinal drugs. And before proceeding to analysis of drugs, we have studied that the drugs need to be extracted from the biological matrices by various methods like solvent extraction, under which we have studied the ammonium sulphate method and Stas-Otto method. After the extraction, now you have got these samples in substantive amount, and then these residues so obtained are further subjected to preliminary and confirmatory, the various color tests we have discussed, and also you can then conduct the TLC and compare with the known sample as reference or control sample, which will be telling you for sure that yes, this particular drug is present in that particular biological sample.